The overall goal of this experiment is to observe production of the chemicals 2,3-butanediol, R-acetoin, 2-ketogluconic acid, and xylonic acid by Klebsiella pneumoniae using the hydrolysate of bamboo as the feedstock.
This method can help answer key questions in the biotechnology field, such as chemical production through biological routes. The main advantage of this technique is that the hydrolysate of bamboo can be used as the feedstocks for chemicals produced by Chemicals-Klebsiella. Demonstrating the process will be JoVE, a pretreated of our lab.
To begin, using a series of sodium hydroxide solutions in 250 milliliter flasks, add 5 grams of bamboo powder to 40 milliliters of sodium hydroxide to make a 10% solution. Incubate the solutions in a water bath at 60 degrees Celsius or 100 degrees Celsius for 60 minutes, or incubate the solutions in an autoclave at 121 degrees Celsius for 60 minutes. Following the incubation, centrifuge the mixtures for 5 minutes. Then remove the supernatant, add 100 milliliters of fresh water, and mix. Repeat the washing 5 to 6 times until the pH of the supernatant reaches 6.8. Then to carry out enzymatic hydrolysis, use 98% sulfuric acid to adjust 50 milliliters of each solution to pH 5.0. Next, add 0.5 milliliters of 200 filter paper activity, or FPU, per milliliter of cellulase. The final ratio